Hello everyone. Welcome to one more video of special Bio-Tonic. Students, in the last lecture we talked about double fertilization process in angiosperm. So on the screen we can see the embryo sac. It shows three cells towards a micropylar end. They are called as egg apparatus, which contain egg in the center and it is surrounded by two cells, that is synogytes. We can see two nuclei in the center. They are called as secondary nuclei. And we can also see three cells towards the chalazal end. They are called as antipodals. Now, we know that in case of angiosperms, the male gametes are non-motile and they are carried by the pollen tube. Now, this process is what called as siphonogamy. Now, when this pollen tube releases male gametes in the embryo sac, the process of fertilization begins. One male gamete fuses with one female gamete to form the diploid zygote. The second male gamete fuses with secondary nuclei to form TPEN, that is triploid primary endosperm nucleus. So students, the embryogenesis begins with formation of zygote. So this is a single cell zygote. This single cell zygote divide unequally to form larger cell and smaller cell. The larger cell is called as the basal cell or the suspension cell, and the smaller cell is called as embryonal cell. Now this suspension cell divide vertically to form several cells to form suspensor. The role of the suspensor is to push the embryo deep into the endosperm so that the embryo can absorb the food of the endosperm easily. The lower embryonal cell remains as it is. Now, further what will happen, the cell of the suspensor which is towards the end is called as terminal cell or basal cell, and the cell of the suspensor close to the embryonal cell is called as hypophysis. This terminal cell forms haustorium, and we can see the cell close to embryonal cell is called as the hypophysis. Simultaneously, this embryonal cell divide mitotically to form 8 cell. It specifically divide thrice to form 8 cells. These group of cells are now called octant. Now octant means group of 8 cells. It forms two tier of cell, lower tier of cell which are marked with red color, whereas the yellow colored cells are nothing but upper tier of cells. So students, this upper tier of cell will form the plumule and cotyledon, whereas the lower tier of cell will form the radicle. Now during the further development, this embryonal cells which are forming octant, they develop to form the heart-shaped structure. In the latter stage, the radicle forms root, whereas the plumule forms the stem, and this cotyledon act as the reserve food material. Students, if you're finding this video interesting, then please do like, share and subscribe my channel. Thank you. Students, this is the structure of dicot seed and monocot seed. As the name suggests, the dicot seed will have two cotyledons, whereas the monocot seed will have single cotyledon. We can see the structure above cotyledon is called as epicotyle axis, and the structure below the cotyledon is called as hypocotyle axis. Similarly, we can also see the seed covered with seed coat. Now we know that seed is formed after fertilization of the ovule, which is covered by seed covering, which are made up of inner and outer integuments. So they are accordingly called as testa and tegmen. In case of monocot seed, we can see single cotyledon, which enclose radicle and plumule. Here we can see radicle is covered by coleorhiza, whereas the plumule is covered by coleoptile. In the upper part of the seed, we can see the endosperm, which is covered by aleuron layer, which is made up of proteins. So students, there's a basic difference between dicot seeds and monocot seeds with respect to the number of cotyledons. So I hope you're clear with the steps involved in embryogenesis. So stay home, stay safe. Thank you.